Welcome to Lab in a Box, a program brought to you by Genes in Space to help bring current molecular biology techniques to your classroom. Today we're going to go over the basics of what's in your kit, how to use each piece of equipment, and how to conduct a successful in-class experiment. We'll break this down into 8 main topics: receiving your kit, introduction to pipetting, PCR, restriction digests, preparing for and running gel electrophoresis, interpreting your results, and returning your kit. Let's get started with receiving your kit and going over each piece of equipment. You will receive your loaner kit in a shipment of 2 boxes. The first will contain the food safety lab kits, which will ship on ice. The contents of these kits must remain frozen until you begin the lab. Make sure you put them into a freezer immediately upon receipt. This kit will contain Easy PCR Master Mix, Food Safety Primer Mix, 4 template DNA samples, a restriction enzyme, nuclease-free water, and a DNA ladder. The second box will contain the equipment needed to perform the PCR and electrophoresis activities. The case will come packaged in a cardboard box. Make sure you keep this for shipping the kit back when your loan period is complete. You will receive a combination to unlock the case by email, and you should have your equipment checklist ready upon opening so that you can assure you have received all of the necessary components. You will receive 8 H20 pipettes, 1 H200 pipette for instructor use only, pipette tips, microtubes and PCR tubes, 4 mini racks, all-in-one agarose tabs and powder to make TBE electrophoresis buffer, Clearview spray and a microfiber cloth, 4 gel casting platforms, along with 8 casting trays and combs, 4 blue gel boxes and power supplies, each with a blue base, buffer chamber, and orange cover, 4 mini-PCR machines, each with a USB cord, power supply, and AC cord, and 4 fold-a-views. You will additionally need to acquire and supply a few things on your own, like distilled water and flasks and beakers. You can find the full list of instructor-provided materials in the instructor manual. Once you have acquired all necessary materials, it's time to familiarize yourself with the setup of the Lab in a Box activity. In this activity, students will examine 2 mock food samples by amplifying a short region of the DNA using PCR and digesting it using a restriction enzyme. They will then examine the resulting DNA fragments using gel electrophoresis. The example scenario is described in more detail in your lab activity packet. The full activity will take about 180 minutes or 4 45-minute class periods. We recommend the following structure. Class 1, Genes in Space and Pipetting. Class 2, PCR. Class 3, Restriction Enzyme Digestion. Class 4, Gel Electrophoresis.